Okay. Do you see my slide? Can you see my slide?
Yes, doktor.
Okay.
Yes, Doctor.
Bismillah. Okay, can I start?
Boleh, doktor.
Okay, Alhamdulillah. Bismillahirrahmanirrahim. We start again. Assalamualaikum warahmatullahi wabarakatuh. So today we're going to continue on another topic on parenteral formulation for peptides, protein, and monoclonal antibody drugs. Okay. So basically, is still in general point of view on parenteral's formulation. However, the preparation and the background of today's topics, we will look at on how the formulation on the production in terms of specifically in terms of proteins. However, still in general forms on the background study and the formulation of proteins. Okay, right. So first we look at the development of biopharmaceutical parenteral dosage form. Right. So on a view, the objective for today, we will look at the overview of conventional parenteral technology and then key factors in considering specific delivery system. And the third one will be the development of new delivery system of biopharmaceutical drugs. Okay, so basically around most of the drug delivery is more or less is parenteral is the major delivery route in delivering drug. Sorry, however, we can say about 95% more that drug delivery will based on parenteral application, okay, parenteral way. And basically the choose of parenteral drug delivery system were done to ensure that the drug will reach a specific drug target area via blood in lymphatic system. So that's why we choose this kind of delivery application. Okay. Next, we look at the key requirements to consider and physicochemical factors. Okay. So first, overview of the products, drugs preparation, and major component in the formulation. All right. So basically, a little bit of background in proteins. Okay, proteins is something a complex compound. Okay, and also a high molecular weight. So basically, protein cannot be delivered via transdermal because of high molecular weight and highly purified. Okay, and high tendency to aggregate. So this is the background of proteins, and the pharmacist or formulator in the formulation of parenteral delivery of proteins must look at must consider this kind of properties of protein to ensure that the delivery or formulation of the whole system before the parenteral, before the injection have been done itself. So it must be take a look at, right? Okay. Next, key requirements to consider: chemical reaction, chemical instability, because the proteins, as mentioned, high tendency to aggregate. In terms of instability, we cannot say that it is less unstable, but it can be the chemical properties of the protein. They can undergo certain reaction. That meaning, this reaction will produce a new type of proteins or new type product. Okay, maybe some kind of breakdown. Okay, due to the instability. So at the end, if this occurs, okay, all the therapeutics properties that been backed by the protein is tidak is not applicable lah. Okay, at the end, because this due to this kind of chemical reaction. So this chemical reaction here, some kind of chemical reaction can occur or usually occur to the proteins. First, we have oxidation in the peroxide with peroxide, and then racemization, conformation like cis strands, because certain in cis strand is very important to look at. Okay, because they are due to some cis strand, even though the compound itself is the same, but the conformation, okay, I think the conformation have been done, and conformation of the compound itself, the certain compound conformation that only will react for certain reaction in terms of bioapplication. Okay, later on you can see in the general chemistry. Okay, in earlier days in your chemistry, it's related about the isomerization. Okay, and then we have the amination. Okay, breakdown of the protein into the amino acid group, and then proteolysis. Again, it's a breakdown of the protein but into a polypeptides. Okay, and then we have photolysis reaction with photo aggregation, and then isomerization. Okay, right. And to prevent from previous chemical reaction can occur, okay, to ensure that the protein can deliver the therapeutic uh, efficiency towards the targeted application. These are kind of stabilizing excipient can be added, okay, towards the formulation to ensure that there is no changes, okay, there is no chemical reaction occur within the protein that want to be delivered. So we have amino acids, surfactant, polyhydric alcohol, antioxidants, phospholipids, and chelating agents. And even though there is some kind of stabilizer excipient that we can choose, again, we must ensure that the excipient that we choose will not react with all the protein, and we will not react all the formulation that have been added within the system. So it's quite difficult actually, okay, to undergo to formulate some kind of new drug or delivery system. Okay, however, because we know that what kind of protein that we want to deliver, we already check the background, okay, that we mentioned in the first slide here. So it's easier for us to choose what kind of excipient that was suitable to be added within the formulation. Okay, these are some kind of example of free formulation physicochemical practice. Okay, the effect of stability, the reaction with pH, temperature, light, oxygen, and then the undergo of polymorphism, stereoisomerisms, okay, shear surface duration, and so on. Okay, this one were related to what physicochemical factors. Okay, must be considered, okay, for protein drug formulation. So within the system during the formulation, we must ensure, okay, we must look at this kind of physical chemical factors before we make any formulation, right? Okay, pre-formulation studies are designed to cover a wide range of properties, so must be in short time and to learn as much as possible, because it must be short, learn in a short time, because take note that this is the formulation that may be that very important, like the commercialization or the use of COVID-19 vaccines that must be uh, as soon as possible, because it's already an outbreak. Okay, and then to learn as much as possible, because we want to ensure that the product or the drug that have been delivered or the use, okay, by the consumers or so on, or the patient, must have the highest therapeutic index with less or with no negative effect. So that's why in short time and to learn as much as possible. Okay, then focus on the identification of potential problem early enough to evaluate potential alternative to stabilize future formulation that could lead to a product. Okay, right. So variable to be tested. Okay, in a protein with ionic strength, pH strength, temperature, inertia, agitation, and surface denaturation. This is related to the stability of the protein. Okay, again, this is due to the, must we look at the physical chemical factors to be considered. Okay, right. Next, the experimental condition for initial population study. Okay, right. Before I continue, any question before I start with experimental condition? Next subtopic. Any question, class?
So final question,
No
Doctor.
question, Doctor.
No,
Okay,
Doctor.
so
No,
just
Doctor.
so previously I say is just some kind of introduction. Okay, thing. Okay, so the parameters in free pre-formulation studies. Okay, in the parameters in formulation, what we will learn today is not that general. Is we can say it's just general point or parameters or point of view that we must look at. It's not necessarily that we look just for these kind of parameters. It just we can say this some kind of initial parameters that must be looked at in pre-formulation studies. Okay, so what is the kind of things that we must to ensure that everything is in good condition before the formulation have been set up. Okay, first is the formulation of the active drug. So the type of drug, the choice of drug that we want to deliver or to want to use, and they have the therapeutic index, therapeutic effects. Okay, first, select an active drug which has as much as characterization as possible. Okay, and then we must know the level of homogeneity and whatever possible, and then lot to lot reproducibility. If any impurities are detected, there should be qualitative and quantitative identification. First, we choose the kind of drug we want to use, and we must sure that the selection of the active drug must be the most efficient enough, okay, to be in the formulation. And then we can say, and then it's not only the 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 efficient of the type of drug itself, however, how it can be produced to lot reproduce, because in terms of formulation, you want to ensure that it is possible to produce one batch, two batch, three batch with the same properties and so on. That's the idea of lot to lot reproducibility, right? Then the characterization of active drug. Okay, it is the paramount importance to a successful formulation project. Okay, physical chemical properties should be elucidated, and to select the most compatible excipient with the active drug and with the other formulation excipient. So we must know, as I mentioned in the introduction, we must know the the properties of the active drug. Okay, you want to ensure that we know all the things about the drug. That's the main. Okay, we must know all the properties of our selected active drug, because from that we can look at the type of excipient, other formulation, other ingredients, okay, like solvents, pH, excipient that to control pH that was suitable to be selected to be included within the active drug. So that's why it is important that we first know what is the properties of the active drug. Either it is the physical properties or either it is chemical properties. Both properties is important for us to get as much uh, as possible the information of this type of drug they want to choose. Okay, all right. Okay, next, the pH range within the formulation, which is must to ensure that because we want to put it, okay, in a human body, specific pH unit will be determined during the final formulation studies 3, 5, 7, and 9. So why it's mentioned by the final formulation study? Because during that our formulation, maybe we add our drugs is some kind of acidic, okay, something like that, and then we put some ingredients, some solvent, blah 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 blah, it become alkaline. So at the end, we must add some excipient to reduce this kind of pH, to ensure that the pH is suitable to be injected, okay, in the human body. So that's the idea why it must be determined during the final formulation studies. Okay. Other than that, some example, maybe all the ingredients that we put within the formulation, okay, our drugs, our solvent, our preservative, our stabilizer, and so on, the formulation is still in acidic form, too acidic. So we put some pH ranger. Okay, so we'll change the pH until the formulation acceptable pH. So that's why it must be determined during the final formulation. We can adjust the final pH, okay, the pH during the final stages of our formulation. Okay, pH changes may have very impact on the solubility and stability of the formulation. pH control in pharmaceutical dosage form is critical, and the proper pH selection is one key factors in developing a stable product. Okay. Okay, next, buffer. Buffer selected must be from USP based on optimal pH range. Okay, we can look at from the USP and FDA approved on the selection of buffer. So buffer concentration must be only used should be range from 10, 15 or 20 micro, right? So as buffer concentration increase, the pain upon injection increase. So these are example of buffer that have been approved by FDA and been stated in USP, okay, US Pharmacopoeia to be used in human body, okay? Right, and then chelating agent. Okay, replication of the active protein residual metal ions could be present acid contact stainless steel, iron, or copper surfaces. This is possible that your proteins that you want to develop can have residual metal ions. So you must put a chelating agent to remove this kind of chelating agent. Okay, the use of a chelating agent is requirement. Okay, recommended dosage around 0.01 to 0.05 to bind or chelate the metal ion present in the solution. Usually, the type of chelating agent been approved is EDTA. At least all of you have heard about the EDTA. So EDTA is widely used as a chelating agent. Next is antioxidant. Antioxidant is one of the major factors in protein degradation. Okay, and specific antioxidants are required, such as ascorbic acid, monothioglyscerol, and alpha tocopherols. And this is the recommended antioxidant dose 0.01 to 0.1% of antioxidant, okay, to be added. Next is preservative, right? Multi-dose formulation require preservative, an antimicrobial agent. The preservative must meet the USP requirements and the antimicrobial effectiveness test. And the most often used preservative for proteins or combinant products are phenols, chlorobutanol, and benzyl alcohol. So usually preservative or antimicrobial agent must be added because the pH of the formulation can grown is the place for microorganism grown. So that's why to ensure the shelf life can be increases, can be increased. Okay, we must add antimicrobial agent. Okay, next is glass vial selection for initial study. So other than the excipient itself, the glass itself, okay, also have important role in formulation start. Okay, when you start a formulation, okay, not only for parenteral but any other type of formulation, type of vial selection is important. So type 1 glass, all experimental work begin with this type of glass, which is a borosilicate glass. Okay, the interaction of the protein with the glass surface should be determined, and glass is not an important is the glass is not an inert material, and glass surfaces must be taken into consideration to study the absorptive properties of the respective protein. Meaning, we must ensure that the type of glass that we use as a vial have no interaction, okay, towards our, especially towards our type of protein that or type of drug that we choose. Okay, in the package selection, okay, glass vial or plastic vial, which is type 1 glass based on USP specification. Okay, we have must can protect our type of drug we choose from moisture, any absorption towards the glass of the protein, and the compatibility of the glass and the protein, or the type of vial, sorry, and then the size requirement in the type of. So meaning size requirement, meaning that if only want to deliver each dose only 1.1 ml, we doesn't need 10 ml punya size lah, right? That's the meaning of size requirements. And then package selection. Package selection is also important. Okay, any other possible form of degradation. Okay, degradation, the structure, the silicate gas, chemical degradation, dehydrogenation, and any kind of absorption towards the package selection. So next, we look at this prober stopper selection. Sorry, I'm too fast, or you can catch up with me?
Boleh doktor. Cuma laju
Okay.
sikit je. Sikit
Okay,
je.
sorry. Okay, right. So next we look at the rubber stopper selection. The composition of rubber stoppers required studies, okay, on compatibility with protein. Again, it's the same as the selection of the glass vial. So meaning we must ensure there is no interaction, there is no, it must be stable, okay, to be used in our formulation. So that's the important things. Okay, in choosing a rubber stopper selection, right, it's possible that particle shedding from the stoppers, okay, into the protein solution via absorption, okay, via, and then same, sorry, absorption and then permeation through the stopper, flexibility of stopper properties. So meaning the particle of type of the drug can move, okay, towards the rubber stopper selection. That's why it is also important to ensure that the particle is remain within our vial. Okay. So elastomic closure. I think you are well known that this kind of upper here is the elastomic closure. Okay. And then the selection of elastomic closure also as the same as the selection of the vial, also as selection of the rubber stopper selection. It must be no reaction and so on. Must be at the end. It must be uh, can be put it, okay, as a whole system. Okay, and the stopper should be essentially non-reactive, physically and chemically, right? The closure with complete barrier to vapor gas permeation. So to ensure, because this type of dynamic pressure closure, some of it can pass the type of vapor and gas permeation through the elastomic closure. So that's kind of other properties that we must look at. Okay, it should have compressibility and resistability. It is important, resistability. Be resistant to coring and fragmentation, and maintain the seal interface and packaging integrity to ensure that the drugs, the dosage that been delivered, okay, in a good form, right? In a good way, right? So next, we look at the membrane filters. Okay, before I continue to the membrane filter, maybe a quick look at every formulation that we must be look at first. Before anything, we must look at we must choose what kind of drug they want to that's suitable for uh, our application. So that's the idea of selection of the drugs. Okay, we must learn as much as possible about it, the chemical properties, the physical properties, and then it's also related about characterization of the active drugs. The characterization, maybe we have some initial study what the active drug that we want to choose. Next, we must know, is it suitable for us? Is it have the characterization or the properties of this kind of the active drug is suitable for our application? So that's why characterization of the active drug is also important. What pH range must be at the final formulation. Either initially is not suitable pH, but at the end we can add some excipient to ensure the final formulation of the pH is suitable to be used in human body. And then the buffers is some kind of solution. The solution must be important to deliver the properties to ensure there is no must be suitable for human. And then that first is not difficult because the list have been given by USP and FDA, either this kind of buffer suitable or not, what pH range is suitable to be used. So we can tally the buffers, the suitable buffers, selection of buffers with the selection of the active drug. And then we must add chelating agents related to the chelating of the whole system, usually the EDTA, and then antioxidant, okay, preservative, if but we can say preservative it's if it is necessary. Okay, if it's not necessary, some kind of like example like COVID-19 vaccines that it must required in a low temperature places. Okay, low temperature is not some kind of place for antimicrobial to grow. So maybe we doesn't need the preservative, is some addition if it's required. Okay. Next, the other than the type of properties within the formulation, the glass system, the glass vial, the package selection, and rubber solution and stomach selection also most important. So at the end, there is no chemical reaction, there is no physical reaction, okay, towards the whole system within the formulation, because we know that within the formulation there is numbers of ingredient we put. So we must ensure that the type of closure, the vial must protect our formulation, and then without uh, put any chemical reaction, or must ensure that the stability of the formulation that we want to deliver. Okay, right. So next, the membrane filter selection. Okay, in the membrane filter selection, all protein formulation are acceptably filled for final sterilization of the product. So the formulation we filter the type of formulation. Okay, that's the selection of membrane filter. So the chemical nature and pH of the membrane must be looked at. Okay. Next, polyvinyl difluoride, polycarbonate, and polysulfate, one of example most compatible with several protein with minimal amounts of protein binding and deactivation. Okay. Membrane filtration, which is the sterile protein peptide product, can only be prepared via sterile filtration to ensure that the filtration process of via the membrane filtration. Okay. Filter media are tested for protein formulation with minimum absorption and maximum compatibility are mixed ester, cellulose acetate, cellulose nitrate, fluorosulfone, and nylon 66. These are some kind of membrane filtration that we use, tested for compatibility with the active drug substance. Okay. Degradation mechanism. Okay. Physical and chemical degradation to be observed and recorded at varying temperature levels. Okay. The formulation must select methods, those that can differentiate physical and chemical degradation quantitatively and qualitatively. Okay. So after all the product have been formulated, okay, we must look at physical and chemical degradation of our formulation. Okay, degradation of our formulation to ensure that the whole system. So at the end, we know the shelf life of the our formulation. So we know that certain time, okay, all the excipient that we put have some kind of expiry date. Okay, at the end maybe some kind of small reaction that occur that at the end that during that range of time it can be occur that at the end will uh, merosakkan our formulation. So the degradation mechanism we must know, okay, at a certain time, a certain temperature, at certain levels. Okay. Next, the product formulation. Okay. The development of quantitative method of testing any active protein is a paramount importance. Meaning, as I mentioned before, at the glycation, after all the ingredient, the type of product have been selected, we already choose. So we must look at other kind of before we put it in, before we release it as our product. So we must look at the petrolysis, must look at impurities induced by oxidation. These are quantitative method that eventually detects, okay, potential chemical degradants and some kind of contaminants. And then the physical instability, the type of aggregation can occur, denaturation, absorption, precipitation within our formulation. We can say the final product of our formulation before we put it in the market, or before we given it to the patient. All right. So analytical method used must be reproducibility available to ensure regulatory compliance for the FDA, analytical methods and bioactivity methods. Okay. So formulation, initial compatibility testing of the active drug substance with excipient tested. Favorable stability factors such as temperature, light, and packaging components. Initial degradation product of the pre-formulation. Performance of stability method indicating the stability indicating testing. Okay. This is also this kind of formulation is also related the product formulation, meaning after all uh the formulation have been done, so before it being into given to the patient, so all this thing formulation background testing must be done to ensure there is no in the long term, in the short term idea, okay, must be acceptable to the system. And then multi-dose formulation, meaning there is numbers of protein, okay, can be within our system. Okay, we have protein A and protein B in our formulation, so that's we call multi-dose formulation. Okay, multi-dose formulation usually take more time for development. Why? Okay, first, several antimicrobial agent must be screened for compatibility with the proteins and the excipient of the formulation. So we must ensure that type of excipient from that's suitable for protein A and protein B. It's not only selection of excipient that's suitable for protein A, but we must know there is no it must suitable with both protein. So that's why it might take longer to develop the multi-dose formulation. So antimicrobial agent are known to be protein inhibitor. Okay, this preservative must be able to kill inhibit microbial that contaminate the product. Concentration of the preservative of antimicrobial agent may inhibit the protein drug, and significant amount of laboratory time must be devoted to this project. Okay, that's why the idea of multi-dose formulation takes some time. Tested to determine its efficacy according to the antimicrobial effectiveness. If the result support the USP requirements, international requirement must then be met. Okay, international market require different preservative, different concentration, and different excipient of the formulation. So that's why uh, uh, to produce a multi-dose formulation is take more time compared to single dose formulation. Okay, lastly is the stability program. Okay, the stability program is not only related toward the laboratory method, but idea of how must be transported and how it can be used and how it be reused. So the stability program, we can say it's related from A to Z, from the laboratory towards the patient. So this kind of stability program must be included, must be take note before we towards our product. So we must look at the robustness of the formulation, characterization of the degradation product, recommended product specification, what type of specification needed, the shelf life, the place they want to store the drugs, and so on. Accountability of physical and chemical variables, and real-time stability data supporting expiration date, shipping tested, design, perform, validated at different temperatures, under shaking and freeze thaw condition. This kind of stability program or done after we already formulate the system. Okay, after we already know what kind of reaction of degradation that can be occur. Okay, here, right? And then afterwards, after we have done with the formulation, anything other characterization of the formulation that we prepared. So this kind of stability program is the program to ensure that it can be delivered specifically towards the patient without any, and we can deliver as much as important about our formulation towards our system, towards our patient. Sorry. Okay, we can say these are all uh, the type of idea. Okay, the background within the experimental condition of the pre-formulation study, the pH, the glass vial, the type of preservative, rubber stopper, antioxidant, okay, buffers, membrane filters, and package. Okay, so these are the steps in development of biopharmaceutical parenteral dosage form. First, we must look at the key requirement of free formulation and formulation before even we start the formulation itself. Okay, and then the pre-formulation physical chemical practice, meaning either what we call a pre-formulation physical chemical, we must look at each type of properties. Okay, either based on the previous study, either this kind of excipient was suitable with this excipient, either this excipient were suitable with our drug. So that's we call the pre-formulation study. And then we done the pre-formulation. Okay, after we have done the formulation, we look at the initial variable to be tested. So the degradation, okay, the absorptionness, okay, the well known, the pH, okay, this kind of initial variable to be tested. And then the last one is experimental condition for initial pre-formulation studies. Okay. Okay, class, any question?
So far okay.
Doktor,
Yep.
satu question.
Yep.
Untuk kalau macam doktor cakapnya pasal ubat ni, if let's say kalau macam ubat tu produk which is the active metabolites dia yang buat kerja, yang give more effect compared to the main parent punya drug, so kiranya dia kena adjust balik ke ataupun boleh just stick sama on the pre-formulation yang dia orang dah buat tu ke?
Usually kita kena tengok balik lah background that kind of yang tu. Usually kita sebab tu dia punya idea dia kita consider dulu. Either it is suitable with the formulation that been done, cuma kita nak change the type of therapeutics tu kan, betul tak?
Betul.
So it's easier for us to formulate the system, but at the end we must take the step by step again lah. 1, 2, 3, this 1, 2, 3, 4, 5. Even though so easier for us because this part is very difficult for us or the pharmacists to develop a new, because there's a numbers of formulation within the system, the SCP and the kind of CPN. So we know that that particular drugs is better from drug B is better from drug A because we want to apply with the same idea, with the same application. So we can look at the type of formulation, the ingredient within that A that we have already been delivered. So we look at the, either it is suitable with B punya protein. If not, so it coming back to square one. Faham tak?
Faham.
Okay. Any more question?
Thank you, doktor.
Alright. Any more question?
No question, doktor.
Tak ada, eh? Alright. Okay. So next we will look at. Can I continue or you want to take some rest?
Boleh je, doktor.
Boleh, eh? Right? Okay. So we look at novel parenteral formulation and alternate delivery system for protein, peptides, and monoclonal antibodies drug. So, alright. So the alternate delivery system may have highly desirable attributes and improve utilities for the clinical administration of novel medicinal and chemical agent. Okay, this attribute included following: targeting delivery to specific tissues, improve safety and efficiency, decrease in the frequency of dosing, reduction of toxicity, and reduction of pain-related administration. So the idea of selection of type of delivery, or the idea of increase the technology of drug delivery were attribute to these properties. Okay, because we have to ensure, at the end we want to ensure it's suitable for the patient, and again it will deliver, it can treat the patient with high efficiency. That's why the idea of choosing a novel drug delivery system, choosing developing some kind of new technology in the delivery system. Okay, so in selection of novel drug delivery system, pharmaceutical company objective are based on this kind of objective to ensure that the development, okay, of new type of the delivery formulation or new type of technology to be delivered to the patient will meet this objective. Okay, must meet this objective to ensure that the product is acceptable. Okay, this kind of product is new but it's not meet that the objective, so no point for that kind of pharmaceutical company to develop this kind of drugs. Okay, so some of key delivery system, we have pegylation, we have microparticles, we have nanoparticles, and we have micelles. This kind of novel drug delivery system. Okay. And then we have osmotic implants, we have inhalation, transdermal, and needleless injection. Okay, this kind of selection of the drug delivery system. Okay, some keys of the drug delivery system. But we can say nowadays all of this is we cannot say it's a novel. It's not novel anymore because previous study has been done, as much as study has been prepared. Okay, numbers of data have been prepared. Okay, due to this new drug delivery system. More or less, this type of new drug delivery system were related toward the suitableness in terms of what kind of application that we want to use, what kind of protein, what kind of drug that we select. Okay, either it can given as much as possible. Is it good for the patient? Is it good to treat the diseases? So that's kind of idea making it type of new type of delivery system. Okay. However, there's a many obstacles to overcome by this technology. First is establishing the physical chemical properties of the active drug substance. Pre-formulation is, we can say it's some the first part, but the most physical, the most it's not hard, but it's take a quite long time, okay, to be in terms of pre-formulation, because we must ensure this kind of excipient is suitable with the excipient. We must have the characteristic, we must have all the properties or within of our system. But the FDA regulation and the USP books can help us to selecting the type of ingredients within our formulation, right? So the third one, determining initial compatibility of the active drug substance with the formulation experience related about the pre-formulations. The fourth one is selecting compatible solvent for the active drug substance excipient in order to establish optimal solubility. Again, the numbers of the buffers, the pH buffers, okay, the solvent, is it suitable? Maybe if it's suitable for the acidic, the protein only suitable for acidic, is it how we want to deliver? So that's why we must that's why the new type of novel drug delivery system have been developed, okay, to undergo to meet these drawbacks. All right. And then the fifth one, developing initial analytical and quantitative assay method that's specific and reproducible, designing and approach appropriate initial stability profile, evaluating and identifying initial degradation product. Okay. And from all these formulation data from 2, 3, 4, 5, 6, and 7, selecting the most promising pre-formulation result. Formulator can be designed, and then the 9-1 packaging selection and compatibility of the active drug substance with formulation excipients, and then determining particle size control, determining appropriate method of sterilization, establishing of reproducing process, ensuring that the product is manufacturable, ensuring that the product meets the specification of required regulatory agencies. Some countries have different kind of regulation, so the idea is different. Okay, so that's some kind of obstacles to be overcome by this new technology. So again, when we developing a novel drug delivery system, we can say it's almost the same as the preparation of the during the pre-formulation. I did suitable. Okay, we cannot say okay, we want to deliver protein A and maybe we choose inhalation. We cannot say like that. So the development is not based on that. So there's a numbers of work to be done, numbers of data to be collected before we select any type of novel drug delivery system that's suitable for our application. Some other problem is that we can prepare some kind of pre-formulation. At the end, we can produce a good formulation. However, we have problem in delivery. Some of developed novel drug delivery system have that drawbacks, that some kind of drawback. The formulation is good, okay, all the excipient is good, the pH is good, but there's some problem with the delivery ways. So that's why it's from A to Z, and the developing of a new kind of drug delivery system for commercialization to be used as inpatient is difficult compared to the laboratory idea or laboratory works, okay, to deliver this kind of any type of new drugs or so on. So in terms of data, we can collect it within laboratory. However, the challenges, okay, towards the pharmacies, towards the pharmaceutical company, is how to make this kind of new type of delivery system to be commercialized. One of other problem is either it is suitable to be in larger batch. Okay, in a laboratory study, we only do it a small lab, small scale. Can be up, can be this kind of new type of delivery system in the upper scales. Okay, higher scale. Is it suitable? Is it can be controlled? Is it can be produced in terms of for commercialization? Okay, I think that's all. Any question, class? Hello, class? Hello,
So
hello?
far no question, Doktor. Tak ada question.
No,
Oh.
Doktor.
Okay.
No, Doktor.
Sekejap. Suat. Kita